Then phone doesn't even have a mute thing, doesn't work. And I'm sorry, like I told you, I took over my husband's office. He works from home. And so his phone cannot be turned off. Okay, so muscle tissue. We learned about epithelial tissue and connective tissue. Here is our chance to learn muscle tissue. So muscle tissue, first of all, there are three types of muscle in the body. Skeletal muscles, which are attached to the bones. We call them skeletal because these are attached to bones. Then cardiac muscle, which makes up the heart, constitutes heart. It's the only place where you see cardiac muscle. Smooth muscle, which is in the wall of the internal organs. And they have their respective function. Skeletal muscle pulls on the bones, thereby it gives us different types of locomotion, movement. Cardiac muscle, of course, is responsible for pushing blood through the arteries and veins. Smooth muscle is responsible for also another type of internal movement, like air through the respiratory tract, food and water through the digestive tract, urine through the urinary tract, different types of movements. Out of this, both cardiac and smooth muscles are considered involuntary, meaning we can't contract them at will. Skeletal muscle is voluntary, meaning can be contracted at will. I want to flex my arm, I can do that. I want to make my heart stop, I can't do that because this is involuntary. I want the food movement through my GI tract to stop right now, I can't do that. It's involuntary. However, can I move my legs? Can I move my arms? Can I twist my torso? Can I move my fingers, toes? Of course, voluntary. So muscle tissue is different from epithelial. We are talking about specialized. Muscle and nerve tissue, they are considered special or specialized tissue as opposed to epithelial and connective tissue. Although some connective tissue can become specialized, like fluidic connective tissue and mineralized bone tissue. So what are the properties of muscle tissue? They have what we call excitability, meaning they're excitable. They can be made to respond to stimuli. All cells can respond to stimuli, but muscle tissue can respond in a far more visible manner. Okay. So pass electricity through the muscle tissue. What would happen? The muscle tissue immediately will contract. Discharge some chemicals on the muscle tissue, like acetylcholine. We'll talk about this. Don't worry about the name. Muscle tissue will contract. So excitability. Second is contractility. It can shorten and exert a pull or tension. This is how skeletal muscles work. The length of the muscle can shorten, which is contractility. No other tissue in the body has this. Extensibility. You can stretch a muscle, extend. Elasticity, the ability to rebound. So after contraction, go back to its original length, elasticity. After being stretched, rebound, go back to its original length. So excitability, contractility, extensibility, elasticity. Once again, skeletal muscles. Skeletal muscles basically have their own set of function. They produce movement. We learned about bones, and I'll talk about some general features and the relationship between the bone and the muscle, and I will explain what's origin, what's insertion. Skeletal muscles maintain our posture because they stabilize the joints, support the internal organs. Think about your ossa coxa. Basically, the ossa coxa is supporting the weight of the internal organs in the abdominopelvic cavity. Regulate entering and exiting of material. like control over swallowing, defecation, urination, etc. Although it's partly true and partly not true. And maintaining body temperature. So this, we want to learn this feature, this property. Again, we'll go over muscle physiology and we'll probably come across these again. The fact that we can maintain an average body temperature like 98.5, that's what most of us go by, the basal temperature. Basically, that's coming from the contraction of skeletal muscle. Every time skeletal muscle contracts, it's going to produce heat. This is why exercising can make you feel warm. Skeletal muscle contractions produce heat. What does this skeletal muscle look like? Okay, so skeletal muscles, think of the skeletal muscles. Again, these will come handy for muscle physiology. So I'll do a quick review when we talk about this in muscle physiology. Skeletal muscles, understand some basic thing about skeletal muscle. Let's say we have isolated a skeletal muscle. This could be biceps brachii. Everybody understands what biceps brachii is. But basically, this is let's say we isolated a muscle. Actually, let me just draw the bones here. Most skeletal muscle will attach to two bones. Some skeletal muscles will attach, in absence of bones, will attach to connective tissue of another muscle or to the skin, connective tissue under the skin. But this is the rule of thumb. Most of the skeletal, they're called skeletal because they attach to bones. Oftentimes they have multiple attachments, at least two attachments, 2 points of attachment. So let's say these are the two bones. And this skeletal muscle is attached to both bones. The way they will attach will be the body of the muscle. This could be any muscle. Body is often called belly. Okay. So the belly of the muscle, the body of the muscle attaches to tendons, which are, remember we studied dense, regular connective tissue. These tendons are what actually attach the muscle to the bone. So they are like the connecting tissue between the muscle and the bone. Muscle tissue, muscle cells directly do not attach to bones. They attach to tendons, tendons attach to bones. What is this tendon made of? It's made of 3 compartmentalized, meaning 3 layers of connective tissue, referred to as epimysium, perimysium, endomysium. Let's take a look at those epi, peri, and endomysium. So I'm going to insert a slide here. Epimysium, perimysium, endo, office themes. Huh? Why is it giving me office themes? Level edit. Hang on, I just want a blank slide. Is that what you're calling level edit? Jeez. Whatever. Okay. So anyway, epimysium, perimysium, endomysium. So let's just draw this muscle again. The 2 bones. They look like dog biscuits, more than bone, that too broken. But anyway, get the point. So here is the belly of the muscle. Let's say we cut this belly of the muscle along a transverse section. So we are looking at the sectional part. So looking at the sectional part here. That's the belly of the muscle. I'm looking at the sectional part. Muscle cells are long. Skeletal muscle cells especially are long. and they run parallel to one another, and they exist in groups. Okay. So connective tissue, epi, peri, endomysium. These three levels of connective tissue, three layers of connective tissue are basically there to separate the muscle cells into groups. So where is the epimysium? Epimysium is basically above the muscle. Light green is what I'm drawing. So let's say after I have isolated this muscle, I am dissecting an animal or a human, and I have isolated this muscle, and I lay my probe or my finger and say, oh, I'm touching the muscle fibers. No, I'm not. I'm touching the epimysium. Epimysium is almost like, think of laminated part. Something is laminated, a book that's laminated. On top of the laminated book cover, if you rub your fingers, are you really touching the paper? No. Are you touching the cardboard? No. You are touching the very thin layer of plastic that is laminating that book cover. So epimysium has a similar kind of relationship with the muscle fibers. It's a very thin layer, but it encircles and covers the entire muscle, basically to protect. Epimysium. Okay. Where is perimysium? Okay, so then we are looking at this sectional end here. Where are the muscle fibers, muscle cells? You'll see that the muscle cells show up as group. And the group is not equal number. This could be 4 cells, 5 cells. This could be again 4 cells. Maybe this here is 5 cells. Meaning they're all running parallel to one another and they're grouped together. So we're looking at this together as here. And there's a layer of connective tissue that's separating this group from this group, from this group, from this group, from this group, and this. So each group is basically encircled by a layer of connective tissue. So this connective tissue that's present between the muscle cells, separating the groups of muscle fibers. This is what we refer to as perimysium. That's it? No, there is another one, endomysium. So if we look at a single group right here, then what's present between the two cells? Basically, two elongated muscle cells look like this. What's present between this cell and this cell and this cell? Again, connective tissue. So each muscle cell is actually isolated and wrapped in its own connective tissue. So connective tissue between the cells of the group. So groups of cells are together. Connective tissue separates the cells within a group from one another. This is what you refer to as endomysium. Okay, so epimysium, perimysium, endomysium. And there is a name for this bundle of cells, this group of 4. Is there a name for this group of 4? Sure there is. This group of 4 or group of 5 or group of 3, group of muscle cells together. Is there a name for it? Yes. The name is fascicle. This is what typically shows up as grain of the meat. If you're cutting meat, what's called grain of the meat. Grain is nothing but fascicles. This is how skeletal muscle is arranged, exclusive to skeletal muscle. So each muscle is made of number of cells. By the way, the muscle cells are called muscle fibers. We'll see that in a second. And those cells are grouped. And groups are separated. Groups are called fascicles. Fascicles are separated by perimycia. Fibers, cells within a fascicle, are separated by endomysium, connective tissue. The entire muscle is wrapped by epimysium. So what we saw here, tendon. What is tendon made of? All three, epimysium, perimysium, endomysium, that extend beyond the muscle and give rise to tendon. And when you study muscle physiology, you will see that why it's like that in skeletal muscle. The skeletal muscles, each fiber is individually excitable, kind of like in electric wire. We have electric wires that might be, electric lines that might be running next to one another, yet each line has to be separated from another just because interaction between them is not allowed. It's going to spell disaster. Same thing with skeletal muscles. So the organization of skeletal muscle is, again, muscles consist of fascicle. Fascicles, let's look at this again. This is a fascicle. This is a fascicle. This is a fascicle. Fascicles consist of muscle fibers. Fascicles consist of fibers. So this is a fiber. This is a fiber. This is a fiber. This is a fiber within a fascicle. Fibers, don't worry about the details because we'll be covering a lot of these again in muscle physiology. But each fiber is like a cylinder. Okay. Each muscle fiber, each muscle cell is like a cylinder, and the cylinder is made of tiny cylinders within. So cylinders within cylinder. Just like a muscle is made of muscle fibers, cells running parallel to one another, every cell, every fiber is made of components that are also almost cylindrical, running parallel to one another. This is why muscle is so heavy in protein. It's all about protein. And then these myofibrils, these are called myofibrils. Each one is a myofibril. So within a fiber, the whole thing is a fiber. Each component, cylinder within cylinder, is a myofibril. Plural, myofibrils, all three. These myofibrils, if we isolate a single myofibril, then each myofibril is made of zones that we call sarcomere. Don't worry about the details. This is basically so that for muscle physiology, it will be a quicker review. And then each zone is made of what we call myofilaments. Myofilaments are made of actin, myosin. There are more proteins than that, but this is just for a quick reference. So that when I say, you can look at the alternate dark and light zone, it actually makes sense. So fiber, basically, this is a single fiber made of fascicles. Okay. Each fascicle is made of, sorry, not fiber, whole muscle made of fascicles. As you can see, each one is a fascicle. It's a fascicle. So every fascicle, now we are looking at, let's say we extended a fascicle and we are looking at fibers within the fascicle. So all these red dots within a fascicle is a fiber. Now we are looking at a single fiber, cylinder within cylinder. This is what a single fiber would look like. There's a membrane component, cell membrane, plasma membrane, and what's inside, this is what we call myofibrils. This light, dark areas, this is what we call sarcomere. Ignore the Z line and all of that. And sarcomere is made of actin and myosin, which constitutes myofilaments. So this is a myofilament. This is a myofilament. They're made of different proteins. This red one and the purple ones, both are myofilaments, and they're made of different proteins. Don't take it any further than this, because all of this basically, all of this goes toward muscle physiology, how the muscle functions at a microscopic level, which we are not studying yet, but we are going to be studying histology. So having this reference that each fiber is made of myofibrils, fibrils are made of filaments, filaments are arranged in zones that we call sarcomere, which gives them a dark and light appearance. So if you look at this, light, dark, light. So each myofibril will show up as light zone, dark zone, light zone, dark zone. So the entire muscle under electronic microscope, entire fiber, not entire muscle, entire fiber will show up as light zone, dark zone, light zone, dark zone. This is one way to identify skeletal and cardiac muscle. So some terminology. Question?
Yes, so you said it was the sarcomeres that are the ones that show up as light and dark as they alternate?
Mm-hmm.
Okay, thank you.
It's simplifying it a little bit, but right now simple is good. We'll study the details when we study muscle physiology. But yes, sarcomere shows up as light zone and dark zone because of the orientation of the filaments. So again, what is a tendon? Epimysium, perimysium, endomysium. Fused together, converge to form tendons. Tendons are always found at the end of the muscle, where the skeletal muscle attaches to bone. And I'm saying the word the muscle, basically, we are talking about skeletal muscle. Tendons connect a muscle to a bone. Sometimes, some muscles can connect to another muscle, along with connecting to a bone. When muscles connect to a muscle connect to another muscle, still they are tendon of a muscle basically converges into tendon of another muscle, then it's referred to as aponeurosis. So when we study biceps brachii, you might see this term, bicipital aponeurosis. Insertion of biceps brachii, bicipital aponeurosis. What does this mean by bicipital aponeurosis? Literally means part of the biceps brachii connects to another muscle. Aponeurosis is that connective tissue junction where a muscle is connecting to another muscle. Okay, so this you can pretty much ignore. I have it here, but it's better not to overwhelm right now because this is microscopic anatomy. Basically know that each muscle cell is a muscle fiber. Muscle fibers have plasma membrane too. And to remember, muscle fibers have multiple nuclei. Oftentimes, these nuclei show up. Actually, you know what? Maybe not completely ignore. Oftentimes, the nuclei show up not at the center. You know, our idea of generalized animal cell, when we looked at epithelial cell, connective tissue, fibrocyte, chondrocyte, we saw the nucleus pretty much stays at the center of every cell. Not for muscle. For muscle fibers, the nuclei oftentimes show up at the periphery, just below the plasma membrane. So the plasma membrane of the muscle fiber, instead of calling it plasma membrane, it's called sarcolemma. Cytosol of the muscle fiber is different than cytosol of every cell, other generalized cell. Is given a name. The term sarco stand for muscle. Sarco, myo, these are all things to do with muscle. Just like cardio for heart. Sarco, myo, you see this term, you know they have to do with muscle. So membrane that surrounds a cell, sarcolemma. Cytoplasm of the muscle cell or cytosol of the muscle cell, muscle fiber, called sarcoplasm, and especially multinucleated muscle fibers. Muscle fibers are multinucleated because they're made, when they're formed by fusion of multiple cells. And the nuclei are not at center because basically this is packed with myofibril, right? Each fiber is packed with myofibril. So the nuclei show up toward the surface, just deep to the plasma membrane, sarcolemma. We'll use this when we look at histology. Okay, this you can ignore. So I have some of these just for reference. Myofilaments are made of actin and myosin, different types of protein. We'll be extensively talking about this in muscle physiology. Right now, just enough so that we can identify muscle fibers. Okay. So 13, 14, actually 12, 13, 14, you can ignore. Ignore. You can ignore. You can ignore. We'll be covering this. I still just kept them just in case anyone is curious. Okay, let's get down to this sarcomere. Basically, sarcomere is, once again, I always find it helpful to draw. So I like to draw as opposed to pointing at pre-drawn diagrams. This is a muscle fiber, myofiber, skeletal muscle fiber. The skeletal muscle fiber is like a cylinder, like I said. Within that cylinder, what is it made of? Just a general idea that sarcolemma, which is the plasma membrane, basically wraps around like any other animal cell. Sarcolemma actually invaginates too and separates the fiber. Okay. And then fiber is packed with, think of cylindrical rod, as if I found a cylinder. I cut the cylinder in parts and inserted partitions, thereby effectively creating 3 segments of the cylinder. This is almost how the sarcolemma is. It partitions the interior of the fiber. And now I took a bunch of rods and I push the first rod through one end of the cylinder. You're following me, right? So I take a metal rod. This is a cylinder that I made or I found. I cut the cylinder into pieces and I put partition in between the pieces and glued them back. So now when I look through, peek through one end, I don't see the other end of the cylinder. I see the partition number 1. I peek through the other end, opposite end, I don't see the end here. I see the partition number 2. You know what I'm saying? Now I take some metal rods, equal length of the cylinder. I take my first rod and I push it through the cylinder, open end of the cylinder, one open end, doesn't matter which end. So I push it, the rod goes and goes and goes, and it hits the partition. I push it, it pierces the partition. The partition is made of thin material. I continue to push it, it hits the second partition, it pierces through the second partition. And now when I look, I not only see the partition, I see this rod too. Same thing. I look through this edge, I see not only the partition 1, I see the rod as well. I do the same with bunch of other rod. I keep pushing. Each rod pierces through the partitions and shows up at the other end of the cylinder. Basically, the relationship between a fiber and myofibril is the same. Fibers are partitions by the plasma membrane, sarcolemma. Myofibrils are like I said, cylinder within cylinder. They are like cylindrical rods made of protein that are present within the fiber, piercing through the sarcolemma. And when I look at each myofibril, I'll notice that each myofibril has it's not uniform, as if each of this rod is painted part dark and light, and then again dark and light, then again dark and light, dark, light, dark, light, dark, light. Okay. This dark light is because what makes up the myofibrils. Myofibrils is made of filaments, which are made of these two proteins. The overlapping of these proteins is what creates some darker zone and some lighter zone. Some places there are two filaments together next to one another. Other places just one filament. This kind of alternate dark and light zone, this is what we refer to as striation. So when we describe skeletal muscle as well cardiac muscle, we say these are striated. Skeletal muscle is referred to as striated. Cardiac muscle is referred to as striated. Smooth muscle, no striation, no such dark light zone. That's why it's called smooth. So striated, basically this is what it means. Striations, alternate, dark and light zone. And there are names given to this dark and light zone, which right now we'll ignore, because our goal is basically just in a histology. We don't have this kind of electronic microscope that will show it like this. This is probably the best muscle slides available in the best university, and the author had access to it. Basically, that's what it is. What we are going to look at is what the slides that we typically have in our lab. So you're going to be able to see some striation. This whole idea of explaining what the muscle fibers look like is so that you can understand what is striation and what it looks like that. Ignore. This is just to show you the different parts. You don't have to learn it right now. If you want to, sure. I have it here so that some students I know they want a little more complete picture. Sure. Just the details of what these filaments look like, what these proteins look like. But we'll be covering this for muscle physiology. Any questions so far before I move on to neuromuscular junction? This kind of orientation, when we say a muscle has contractility, skeletal muscle contracts, basically the contraction is happening at the level of this sarcomere, right there. This is the sarcomere. Middle of a light zone to middle of another light zone with a dark zone in middle. So half light zone, a dark zone, half of a light zone. Basically, that's what sarcomere is. We'll go over the details. I don't want to go over M line, Z line, A band, I band right now. We're just calling them striations right now. Contraction is happening at this level. Interaction between the filaments, the myosin, the actin, that's basically how contraction happens at this level. So when we observe muscle contraction, you can, yes, overall you can see that the length of a muscle got shorter, but at molecular level, you can actually see at the sarcomere how the filaments are interacting with one another and how contraction is happening. So we'll be studying muscle contraction when we study muscle physiology, and we'll talk about all of this. 144 is a little asynchronous, lab and lecture wise. Just because there's no cell physiology lab, we can't really do much. So it would seem like the labs are way ahead of the lecture. 145 is just completely synchronous. Cardiac anatomy, cardiac physiology, circulatory anatomy, circulatory physiology. 144, when we get into nervous system, that's when the lecture and lab kind of tends to fall together. Any questions so far? This is just to give you a reference again. Otherwise, it just becomes too abrupt. We will be looking at neuromuscular junction. So let me explain that. Skeletal muscles contract because nerve cells make them contract. Neuron, all of you know, is a nerve cell. Fiber, in this case, is a muscle cell. We are talking about skeletal muscle. So skeletal muscles contract, not by themselves, but because neurons tell them to contract. We just started talking about signal transduction. So this is a good place to talk about this. How does a neuron make a muscle fiber contract? It's a type of signal transduction. Remember yesterday we talked about juxtacrine, paracrine, endocrine. Basically, neuromuscular junction is a junction between a neuron and a muscle fiber. The way it works, I'm going to insert a slide here. The way it works, so neuromuscular junction. The way it works, that a single neuron, which looks like this, like a tree with many ends, terminals, typically controls number of muscle fibers. And I drew this for convenience. In reality, it would be N muscle fibers, muscle fiber, muscle fiber. Skeletal muscle fibers are parallel to one another. So when this neuron commands via paracrine signaling, communication, chemical transduction basically, these fibers, 1, 2, 3, 4, will have no choice but to contract. That's the relationship between neuron and skeletal muscle fibers. So this junction between the neuron and the fiber is referred to as neuromuscular junction, abbreviated as NMJ. So this is a neuromuscular junction. So is this, so is this, so is this. Paracrine communication happens here. So basically what the neuron does via a complex process, which we'll talk about later, the end of the neuron that's next to the muscle fiber, it looks like a button. It's swollen, almost a button-like, knob-like structure. This is filled with chemical. What kind of chemical? The chemical is known as ACH. You don't have to know it, but that's what this chemical is. So when this neuron releases ACH, typically it will release it at the same time from all of its ends into the muscle fiber. This is what we call paracrine communication. This ACH, remember yesterday I said neurotransmitter? Neurotransmitter meaning those chemicals that are discharged by neuron over a short distance. Paracrine is short distance. Muscle fiber and neurons are right next to one another. They are not far. So the neurotransmitter, ACH, is a neurotransmitter. Okay. So neuron releases neurotransmitter. In this case, ACH. You can ignore ACH and just call it neurotransmitter. Neurotransmitter just travels a little bit of distance and binds to the muscle fiber. So now the neuron is generating the signal. Muscle fiber is the recipient of that chemical signal via the neurotransmitter. Neurotransmitter is the chemical signal. And muscle fiber will respond to that signal by contracting. It's a prime example of paracrine communication, short distance chemical communication. How do cells signal to one another? How do they communicate to one another? Well, this is one example, paracrine. A cell, neuron, discharging a chemical, neurotransmitter. In this case, ACH. That neurotransmitter travels a short distance, not long distance. Paracrine is always short distance. Binds to the recipient cell, who's a muscle fiber in this case. So muscle fiber is the recipient of the chemical, neurotransmitter. So neurotransmitter binds to the muscle fiber. Muscle fiber responds to the neurotransmitter, this chemical signal, by contracting and shortening in length. Lots of things happen as a result of that contraction would happen. So those steps, we'll learn about them later. So this is an ideal example of paracrine communication, chemical transduction, electrical juxtacrine, short distance chemical, paracrine, long distance chemical, endocrine. We'll talk about that. Any questions so far? So when we look at histologically, these buttons like structures, these neuromuscular junctions, they show up as a long thread-like structure, which is the end of the neuron. This is this. And the junction itself. So in histology, we have some slides where you would be able to see this. This is what we refer to as neuromuscular junction.
Professor,
Yeah.
I think I'm in like a greater detail. So the neuromuscular synapse is literally where it connects. Is that technically the same as the neuromuscular junction, or is it just a part of the neuromuscular junction?
It's technically the same as neuromuscular junction. I'm just calling it junction so that we don't have to explain synapse right now and overload you with the different parts of chemical synapse.
Okay, thank
synapse, you. Yeah, synapse is a name, physiological name of a junction between two cells that are somewhat special. So,
Professor? Oh,
yes.
sorry.
Go ahead.
So the name of the button is going to be the neurotransmitter, right?
No, the name of the button is actually axon terminal. I'm just cutting down some terminologies here.
Okay.
Axon terminal is the button. Junction is junction, basically the part of this and part of this. And if you're wondering if this is called axon terminal, is there any name to the part of the skeletal muscle fiber next to that axon terminal? There is. You don't have to know it for this exam, but FYI, it's called motor end plate. So this is axon terminal, the part of the muscle fiber where the neurotransmitter binds to. That's what's referred to as motor end plate. When you say neuromuscular junction, you are referring to this axon terminal and this motor end plate. It is the junction. Two different parts of two different cells forming a junction without actually touching one another. That's what a synapse is in this case. There are many types of synapse. I just don't want to get into that right now. So what we're looking at here, this is a combo of axon terminal and motor end plate. This is a combo of the axon terminal and motor end plate. If we had electron microscope, that will show the separate terminal and motor end plate. Okay, so there are many types of skeletal muscle fibers. Again, we can ignore this slide right now. I just had them part of the file for those that want to have like a more complete picture right now, but we'll be covering all of this later on. I'm just trying to cover as much as we need to know to understand the histology. So all our skeletal muscles are made of a mixture of three different fibers. And we can leave it at that. Some look red, some look pink, some look white. That's about it. And this is really, for those that are into exercise physiology, a comparative, comparison between the white, the pink, and the red. I just thought that information is cool, so I added it. But for the anatomy exam, you can safely ignore it. Question. Okay, now some terminology that would really be helpful when you are identifying the muscles. which will start from next lab forward, onward. Muscles, skeletal muscles. When we say muscle terminology, really we're talking about skeletal muscle. So skeletal muscles have at least 2 attachments. Sometimes they have more, but at least 2. The attachments are called origin and insertion. Origin, insertion. So what does it mean by origin? The attachment point when the muscle is all from the muscle contraction. So let's say again, my doggy bone here. Gosh, that doesn't even look like a doggy bone, but use your power of imagination. That would come handy in physiology too. And here you got the muscle attached to via tendon. So when the muscle contracts, bone number 1, bone number 2. When the muscle contracts, both of the bones do not move. One remains stationary, the other moves because of the contraction of the muscle. The direction it pulls from. Okay. So origin of a skeletal muscle is the attachment to the stationary bone. If bone number 2 is moving when this muscle contracts, then this muscle's attachment to bone 1 is its origin. This muscle's attachment to bone number 2 is its insertion. Point of muscle attachment that's movable. So let me show you a little bit of, let me just start the video so that I can do a little demo here. Okay, you guys can see me. You can see my arm, right? So everybody, I like to use this muscle, biceps brachii, because everybody knows where that muscle is. So biceps brachii. Biceps brachii has more than 2 via an aponeurosis, but simply speaking, got 2 points of attachment. One is to the scapula, other is to the ulna. It has more than that, but I'm just simplifying. Okay, so we got scapula, ulna. Biceps brachii, when it contracts, so I'm flexing my arm. So biceps brachii, when it contracts, basically that's what I'm making it do. I'm stimulating the muscle to contract. When biceps brachii contract, the scapula doesn't move. However, the ulna does. So when we say what's the origin of the biceps brachii, it would be the what?
At the scapula?
Scapula.
The scapula, and the insertion would be the ulna.
Ulna.
Again, there are specific places in the scapula and ulna, but in the exam, if you can tell me that which bones the muscle attaches to, that's pretty good. Sometimes you have to be a little more specific. Other than that, I'm looking for bone. Okay. So origin, insertion, action. So basically, now the action is what happens when the muscle contracts. The function of the muscle when it contracts. What happens? What is biceps brachii doing here? I'm flexing it's moving my forearm, right? In a flexion movement. Moving the forearm. Flexion of the forearm. So origin, insertion, action. Each skeletal muscle has a stationary bone attachment, origin, attachment to a movable bone, insertion. Oftentimes, I would say oftentimes, but not so oftentimes, there are multiple origins, multiple insertions, at least 2 attachment, 1 origin, 1 insertion. And when the muscle contracts, what happens? Something moves. Basically, that's what the muscles are for. Skeletal muscles are there to produce bodily movement. Something else, just because a bone serves as origin for one muscle doesn't mean it can't serve as insertion for another. For example, I'm just going to turn a little bit so that you can see me here. So here we got groups of muscle, 2 muscles known as rhomboids. Okay, rhomboids major, rhomboids minor. Rhomboids, they basically attach to scapula, and I'm just going to turn this way. Can you see the back of my shoulder here? What I'm
Yeah,
trying
we
to
can
do?
see.
Okay, so basically, rhomboids are muscles that are, they hurt my back. It's proving a little more inconvenient than I like to admit. Right there, rhomboids are right there, right there. So basically, when rhomboids move, when rhomboids contract, they are attached to scapula and the vertebral column. Scapula and vertebral column. When rhomboids contract, it moves the shoulder like this. So basically, the scapula moves. Lateral rotation. Basically, the scapula moves. Vertebral column doesn't. So the same scapula that served as an origin for biceps brachii becomes insertion for rhomboids. So a bone can serve as origin for one muscle, but insertion for another muscle. It all depends on if that bone is moving when the muscle contracts. If it's moving, it's just insertion. Is it remaining stationary? If it remains stationary, it's origin. So understand the origin, insertion, and action. Something else, a trick to kind of remember the origin and insertion. Follow the direction of the muscle contraction. If you follow the direction of the muscle contraction, you can pretty much easily can tell the origin and insertion. It's especially for longer muscles like biceps brachii. If you follow the direction, you know what that muscle does. The direction of the contraction is basically pulling the bone, pulling the bone is inserted to. And if I want to talk about this, agonist and antagonist, prime movers and antagonists. Yeah, okay, actually this might, when you are reading through the muscle action, this might be helpful. So skeletal muscles, they are grouped. No muscle works alone. Skeletal muscles typically work in group. So groups of muscle together do this function, and another groups of muscle together do that function. So prime movers, when two muscles together are doing the same thing, same particular movement, they're producing the same movement, then you say that these two are prime movers, meaning these two are working together to produce a certain type of movement. Again, this is simplifying, but the same thing. You got biceps brachii, that's helping with the flexion of the forearm. You got something called, another muscle called coracobrachialis. Coracobrachialis, which is also working toward when the forearm flexes, that muscle is also contributing to the same movement. So when we say when the forearm is being flexed, biceps brachii and coracobrachialis are working together as prime mover. Again, it's simplified, but you get the point. Antagonists are muscles that oppose the action of another muscle. So for this movement, the flexion of the forearm, if biceps and brachialis are together working as prime mover, which muscle is basically going to oppose that action? Which muscle, when it contracts, will stop the forearm from flexing or from moving? If you're looking at the arm, just based on simple knowledge of where some of the muscles are. Which is that muscle? When it contracts, it can stop the flexion of the forearm, thereby oppose the function of the biceps. Biceps brachialis are going to do this. What is that muscle? Triceps. When triceps contract, watch what happens. So this is what biceps does. Flexion. This is what triceps does. Extension. Opposing the function of the biceps. You see? I'm contracting. Basically, I'm stimulating the muscle by putting pressure. And I'm contracting the biceps. Flexion. Antagonist. Triceps. Opposite movement of what the biceps did. So if biceps and brachialis are prime movers together, you say triceps is antagonist to biceps. And this is necessary. Basically, you'll later on see the antagonist, how that helps. This is how skeletal muscles always work. A group, few muscles, together may achieve a function. There will always be an antagonist that opposes that function. This is to prevent, overflexion, overextension. Same thing. If triceps basically produces extension, what causes or what stops, what inhibits the overextension is the biceps. It resists the action of the triceps.
So would the terminology then be respective, to them in their own right? Is it kind of like relative maybe then? Can they both technically be prime movers depending on what perspective you're talking about and the antagonist?
Which function you are talking about exactly? Now, if you're looking at, it's all about the action. If the action becomes extension, then you can say, okay, is biceps and triceps prime mover in the extension? No, they are not. In that case, triceps and another muscle could act as prime movers. And biceps becomes the antagonist. Yes, so based on the function, a prime mover can be an antagonist depending on what function it is. So muscle X does this function. Muscle Y does that. It moves the bone, X moves the bone this way, Y moves the bone that way. X and Y are antagonistic to one another. So when I'm saying for the function, which is, this case, flexion and extension, opposite type of movements. So this movement here, who's the prime mover? Well, Y and maybe Z, which works together along with Y. X is the antagonist. When I'm looking at this movement, in this case, flexion, who's the prime mover? X, maybe A. Whichever muscle is working together to produce this movement, they are prime movers. So in a way, when you say, is it relative? Relative to the function, relative to the movement you are talking about. So yes, relative to the movement.
Thank you.
And other times you can just ignore fixator. Other times there are muscles called synergists, which assist the prime movers. It will make a lot more sense when we are talking about the actual muscle physiology and some of the functioning there. So prime mover, antagonist, those two are good. I would say synergist and fixator. Right now, you can ignore that. Well, actually, I do have some. So we'll talk about the muscle contraction later on. Isotonic, isometry, where we'll come across synergist, fixators. Let's keep ourselves to prime movers, antagonist right now. This also, it's just if you want to understand the skeletal muscles prime mover antagonistic function. Muscles basically, they work kind of like how the the tools work, how levers work. I would say we can ignore this right now as well. Okay, what else do I have here? Oh, now I actually have muscles. Okay. These are diagrams of the different groups of muscles that I have here. We'll be looking at this. I had this for reference in case you are just using this slide. I just have this for reference. Okay. So oftentimes skeletal muscles, for convenience, and by the way, skeletal muscles, their naming system is more complex than it has to be. Just so that you know, there has been a movement for almost a decade now to simplify the names of the skeletal muscles and basically have them have 4 categories with the names being repeated in those 4 categories. It will just simplify the naming system. But guess what? Most anatomists haven't agreed to it yet, I guess. So we'll talk about axial and appendicular musculature. Just like axial skeleton, appendicular skeleton, muscles of the head and neck along the vertebral column, along the torso, typically they are grouped as axial muscles. And of course, the appendicular musculature, which are attached to the appendicular skeleton. Move the appendicular skeleton, pectoral girdle, upper extremity, pelvic girdle, lower extremity. For convenience, instead of learning these as axial and appendicular, when we go over the muscles, next class, I have basically divided that into muscles of the head, muscles of the neck, muscles of the chest and abdomen, basically again axial muscles, and then we have the muscles of the extremities. So if you look at the lab manual and or the list that I provided, you see that I have muscles of the facial expression, group 1, muscles involved in chewing, these are all axial muscles, muscles of the scalp, muscles moving the head and cervical spine, muscles of the vertebral column, abdomen, so group 1, 2, 3, 4, 5, then group 6, these are all basically axial muscles. Then I have appendicular musculature, 7, 8, 9, 10, 11, and 12. I just have them separated like that, but basically it's axial and appendicular musculature. Any question that you can answer about this? Okay, so let's look at histology. I want you to take my time to talk about the muscles here. Let me pull up another file. Okay. So what we want to do, we have downloaded the histology file. I thought I'll go over the head and neck today, but basically I'm just going to look at muscle and identify muscles with you guys. So next class, we're just going to look at muscle models. It would be helpful if you look at the groups as they're listed in the lab manual or the list I provided, 1, 2, 3, 4, 5. We would probably be going over the first 5 groups or 6 groups of muscle next class. For histology, we are going to identify some basic structure. Again, with the slides that we typically have access to, we don't see a whole lot. So muscle types, skeletal, smooth, and cardiac muscle. I would have you look at some structures of smooth muscle and cardiac muscle, just to compare. So again, skeletal muscle, we know they are voluntary. They have peripherally located nucleus. They are multinucleated. They are striated. They attach by tendons to bone. Their primary function is contraction and move bones, basically, which is why they have origin, and insertion. So again, this is just to show you, this is probably, it doesn't even look like a real slide. It more like an artist representation. I just have it as a reference. This so nicely shows the perimysium, the endomysium, and the muscle fibers, what I was drawing, the fascicle. And the same thing again. This kind of slides will probably never get a chance to look at. Okay. For us, what I'll be testing you guys on will be this. This is what we actually get to look at in our lab. This is a slide from our lab. But even in this one, you do see certain features. First of all, how do you know this is muscle fiber and not any other cell? Look at the cylindrical fibers. They are long, cylindrical, very long. You can still see that. I'm just tracing the fiber here. Basically, that's a fiber. This is a fiber. Muscle, you don't just say fiber, say muscle fiber. What else can we see? We can see the peripherally located nuclei. So these dark dots that you see here, this, here, here. These are all peripherally located nuclei. Periphery, because they are along the periphery. This is the border of the muscle. These are nuclei. So I'll ask you to identify a muscle fiber. I'm circling those that you need to identify. I would ask you to identify nuclei. What else can we see? We don't really see the striation well yet, unless we magnify this. Fiber and peripherally located nuclei. Just by looking at the nuclei and the shape, you can still identify that this is a muscle fiber. More than that, to identify the tissue. What kind of tissue is this? Would you just say muscle tissue? No, you actually say skeletal muscle. How do you know it's skeletal muscle? Well, because only skeletal muscle has fibers that run parallel to one another without being connected to one another. If you look at this, these are running parallel to one another without being connected to one another. Each fiber is on its own. Look at this. No connection between the neighboring fibers. So even at this is very basic type of slide. You can still identify the tissue as skeletal muscle. I will ask you to identify the tissue, what type of muscle it is. So identify the tissue, identify nuclei, identify fibers. So this is basically a slightly better stained version. You can kind of see the striation. Let's enlarge this. Or into this. And enlarge this, and we actually see this in our slides in the lab. This is from, well, this is not from our slide, but I have seen similar things in our slides. So basically, this shows you a neuromuscular junction and striation. Okay, can we see the striation? What happens in this, in many of these slides, the fibers are actually kind of collapsing together. You got multiple fibers that are staying together just because the way the tissue was moved around when the slide was made. That's why these are not the highest quality slides where every fiber is present by itself. The slides cost way more money than what we can afford. So in order to see the striation, you kind of have to look at individual fibers and see, can I really see striation? It's not going to be visible all throughout, at least not clearly. So let's look at the edge here. Let's focus on this one here. Can you see that how this fiber shows up as a dark, light, and dark area? You see that? Or here you can see that dark, light, dark, light. This one shows it the best because it's a single fiber. See that? Basically, when you say striation, this is what we are referring to. Dark, light, dark, light, dark zone. You can kind of see here too, dark, light. This is just multiple, more than one fiber that got moved together. Light, dark, light. What else can we see here? We can see a neuromuscular junction, kind of like what we looked at earlier. What is this flashing thing? So basically, the axon, there is an axon. Let's follow this one here. Another one. What we look at here, here, here. Basically, this is the combo of the synaptic knob and the motor end plate. You see the dark circle, dark patch like this. You can follow it here to the end of the neuron, what I was drawing, the end of the neuron with axon terminal and motor end plate. Basically, that's what it is. Axon terminal, motor end plate. Is that part of the neuron? Part of the neuron. Axon terminal, motor end plate. Part of the neuron. Axon terminal, motor end plate. Let me use a brighter color maybe. Same here. Basically, each of this dark circle or dark patch that we see here stands for neuromuscular junction. So that's the neuromuscular junction. That's the neuromuscular junction. So is this. So is this. So we got striation. We got neuromuscular junction. I'll ask you to identify neuromuscular junction. And sometimes they come in different okay. Different stain. It's a different stain. I believe this is a slide that I borrowed from, or we borrowed from some other place just to take a picture. But the same thing. End of the neuron, neuromuscular junction, end of the neuron, neuromuscular junction, synaptic knob and motor end plate, part of the neuron, neuromuscular junction, part of the neuron, neuromuscular junction. I'm just abbreviating as NMJ. Neuromuscular junction often is abbreviated as NMJ, but for the exam, I would like to see neuromuscular junction, the full terminology, please. That's what we have for skeletal muscle. So what did we identify for the skeletal muscle? This is very basic. Nothing to be scared of. I know an average student doesn't like histology. And these are muscle histology is not my favorite, just because the quality of the slides that we have access to is so, lack of better words, pathetic. You don't really see a whole lot. So we are identifying muscle fiber. We're identifying nuclei. We're identifying the tissue itself. And we are identifying neuromuscular junctions and striations. So 5 things. Identify fiber, an individual fiber, basically an individual fiber right here. Identify the nuclei. These are stained for neuromuscular junction. I'm not sure if it's a little too dark for the nuclei. So I'll be using, some of these versions, what we are looking at today. Identify the tissue, identify striation, identify NMJ, muscle fiber, and nuclei. Very basic. Come on. Any question? Okay, so we would look at smooth muscle just so that you know what it looks like. In 145, we look at smooth muscle a lot. So smooth muscles, they are involuntary, no striation, no multinucleate, no branching. We're using the word branching because skeletal muscles, they are parallel to one another. You will see in a second, cardiac muscles are actually branched. They are interconnected. Smooth muscle, not branched, no striation, no multinucleate. And smooth muscle looks almost like a dense, uniform, and it takes light stain because there is no striation, dense, uniform, pink, solid tissue. It's very distinguishable, especially when we look at the wall of different organs. In 145, we look at blood vessels, we look at GI tract, we look at the reproductive tract, and we see smooth muscle everywhere. And it's always looking the same. It's almost like a very consistent, uniform, dense type of tissue that doesn't show a lot of stuff. So ideally, again, this is an artist's representation. Ideally, smooth muscle fibers. The cells are not cylindrical. You can see a single nucleus in the middle of the cell. The cells are somewhat elongated, but they look more like this, as opposed to skeletal muscle fiber that we just looked at. So what's drawn here is what we're looking at here. Centrally located single nucleus. I would ask you to identify the nuclei. This uninucleated cell, and you can kind of make up the shape too. Oh, not really. But you do see the uninucleate cells. And identify the smooth muscle tissue, not from this, because this is like more like an artist diagram, but looking at the actual smooth muscle tissue. Like this. This is the wall of GI tract. Or as wall of blood vessel. This is a vein. This is what I was talking about. The smooth muscle looks like a consistent layer of tissue that doesn't show a whole lot of stuff. So this is smooth muscle. This is also smooth muscle. Something else to remember. Smooth muscles, when present in the wall of the organ, typically go as this way and this way, circular and horizontal layer. I know you really can't distinguish here a whole lot, but that's what we have. So if I were you, I would basically identify smooth muscle as, oh, this is part of the wall of an organ. Kind of taking shortcut, but like I said, muscle histology isn't my favorite. I love histology, but we just have access to such pathetic muscle slides. This is the wall of an organ. You don't have to know GI tract, but this is basically part of the GI tract wall. This is blood vessel. Internal organs. The wall of internal organs are always made by smooth muscle. Vessels, stomach, small intestine, wherever there is muscle, wherever there is contraction going on, smooth muscle. So this is clearly the wall of an organ. This is actually blood. So basically this is smooth muscle all throughout. So I would ask you to identify the tissue, smooth muscle, and for lack of better resources, it's best identify as part of an organ. The smooth muscle is the only muscle that forms the body of an organ. Skeletal muscle doesn't. Cardiac muscle only forms the body of the heart, nothing else. I'm going to encircle that. I'll ask you to identify smooth muscle. This is just. And then we got cardiac muscle. Yeah, our cardiac muscle slides were really bad. Cardiac muscles are branching, meaning they are interconnected. And they're interconnected by something called intercalated disc, also known as gap junction. And they are also striated. So I downloaded some free, copyright-free muscle tissue, cardiac muscle pictures from the web, just because our slide pictures were pretty much not up to the mark. So let's look at this here. So basically this at lower magnification, this is the same slide that's being projected at higher magnification here. So this looks very much like skeletal muscle fibers. Just the strain is slightly different. But what we looked at for skeletal muscle fibers, see, this is very similar to that. Except that you can clearly, I'm going to trace it here. You can clearly see that these muscle fibers are joined. So I'm going to track this fiber here, here, here. Notice that this and this fiber, both are joined because cardiac muscle fibers are branching. They're interconnected. Now let's follow this fiber up here. See that? It's joined. Similarly, let's follow this up further, this fiber up further. These two joined. Then let's follow this here. Joined. Pick up here again, joined. So basically, cardiac muscle fibers, unlike skeletal, let me show you that. Gosh, forget that I hurt my back. Let me show you the skeletal muscle fiber. Notice that these are not joined. These are not joined. Looks like several of them have fallen together, but they are not joined. There's nothing branched. These are individual fibers. Or better yet, let's look at this here. We don't see branching. If you think like, oh, isn't that branching? No, look further. This fiber is ending here. Individual fibers, individual, 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 individual. You can see that these are parallel, 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 parallel. This. These are parallel. As opposed to this here, branched, branched. It's the same fiber that is continuing on. It's the same one that's continuing on here. Branched, 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 interconnected. And at higher magnification, you'll see the fibers are joined by this dark line-like thing here, here. This. Even at lower magnification, this, here, see that? And you can kind of make out the striation too, that is striated. So, single nucleus, uninucleate, not multinucleate, uninucleate. And this junctions and the branching, branched. So this branching and the interconnection, the connection between two cardiac muscle fibers via, I'm going to erase this so that you can look at the dark line, as if somebody has drawn the line here with a pencil. Here. This is what we refer to as intercalated disc or gap junction. Intercalated discs or gap junctions. You can use one or the other terminology for the exam. I'll ask you to identify that. As usual, the nuclei, single nucleus, nuclei. And please write nuclei if I'm pointing at multiple of them, not nucleus. And gap junctions. Or intercalated discs. And the tissue. Cardiac muscle. I'll ask you to identify the tissue. Any question? Like I said, this is not like the histology that you have seen before. This is why muscle histology is in my favorite, just because we don't see a whole lot, but it's what we have. It's better than nothing. But later on, the histology gets better. 145 histology is good. Histology is a good subject, by the way. I know most students don't like it. But this is one branch of anatomy, probably the single branch of anatomy that really allows you to learn skills like deductive reasoning. Because it's not as visual. And you really have to think about, could it be A? Could it be B? Could it be C? If it's A, why is it A? If it's B, why do you think it's B? If it's C, why do you think it's C? Histology, if you learn it like that, why do I think this is? Instead of, oh, she just pointed, I remember this figure. I mean, when I was, initially I started college, that's what I would do too. I had few of us, we thought we were so smart, that we could just cut corners. You would remember pink slide with purple, purple slide with yellow, blah, blah, blah. And we did okay. And that too, it's a brutal system. The exams were beyond brutal. You have no idea. When whoever tells me that these exams are hard, and my first response is, you have no idea what hard is, really. I mean, 30 slides and each lab exam is like 3 hours long. And there's like multiple subjects, and histology is just one of them. And it could be 30 slides that you have looked at throughout the year, one exam in the year. And the way the histology is done is you look at a slide, you are given 3 minutes, you have to identify, you have to draw a picture, you have to give at least 2 to 3 characters telling you why do you think what it is. And then you are giving like, I'm forgetting, 2 to 3 minutes, and there's a bell that would ring, an alarm, like more like, move to the next one. Anyway, so much pressure. But still, so part of that, part of the reason it was too stressful, and I kind of tried to take the shortcut, and then I realized that shortcut won't work. You really have to sit down and think about, why do I think? Why are we calling it cardiac muscle? What is it that I can see here that will tell me it's cardiac muscle? So when I'm teaching, I'm lecturing from that perspective. So if you sit down, I know it's all image, online, so it's easier to just memorize the picture and, oh, this is what was pointed. But then you would be missing the skill set. So if you really think about, what am I looking at? Why is it being called cardiac muscle? What is it that I can see? Oh, I can see these lines right there. I can see a single nucleus. I can see branched fiber. Which tissue has these characters? Cardiac muscle. So therefore, this must be cardiac muscle. Deductive reasoning. If you practice that, your skill set becomes that much better. So any question? That's for our muscle histology. Next class, I know I'm just 1 week ahead of you. So by the time next Tuesday comes through, I'll have another file uploaded into the module, which would be basically the model pictures of all the muscles of the chest and abdomen and back, basically all the other axial muscles. I already have the head and neck. And then I'll be uploading the muscles of the shoulder, pectoral, pelvic girdle, and the extremities. So we got 4 classes before the next lecture exam. Next 3 classes will go over the muscles of the head and neck, probably part of the torso. The class after that, rest of the torso, probably the shoulder, the pectoral girdle and the upper extremity. And the last class, the pelvic girdle and lower extremity. Any question I can answer? Okay, so I'm going to stop sharing here. If there are questions, then I can answer now. Okay, so should I consider that we have this covered? And I'll see you tomorrow for physiology. So unless there are questions, I'll see you tomorrow